is not accomplished in a very strong form in a child that ends up with autism. And so this consideration of the nature of these processes as they sort information and as they set up these more complex operations in the brain is crucial and a crucial aspect of understanding what goes wrong in autism. Now, to understand a little bit about the shaping, let me describe just a few simple examples. A child is born into the world, and in the family, in the environment, everyone is talking in a particular language. And of course, from the point of view of the child's brain, the child can't know what environment it will have been born into. You could say, and nonetheless, within 2, 3, 4 months, the brain has created, by sorting the information that are specific to that acoustic experience, an almost ideal processor has been created to pick off the sounds that relate to that native language. And of course, it doesn't matter whether the child's been born into Spanish or Mandarin Chinese or English or whatever. Doesn't matter. The brain doesn't know what language it's going to be what challenge it's going to be presented with, but it sorts sound by natural competitive plasticity processes. And basically, that sorting is the underlying basis of creating an organized brain that can receive language. None of that sound initially has meaning to the child. The child doesn't put meaning to words until late in the end of the first year of life in normal development. Of course, we know that the development of this sorting, the accomplishment of this sorting, and the progressive use of this information in creating language is a fundamental problem that characterizes a child with autism. Now, at the same time, the brain is also sorting information, organizing itself as it's creating a self-referenced identification of the person that is been born into the world. You could say it's also contributing to the genesis of the person, the self, and to the identification of its operations as coming from that self. And this is all based upon the association of the baby with its actions to their source. This is a neurological process, and of course the source of every movement, every feeling, every thought through association is the individual. And that's the basis of the genesis of self. It's at the heart of the creation of social operations, social skills and abilities. And of course, things have to go right in this early period, just as they do in speech reception, just as they do in organizing the brain and sorting information so the brain can control facile movement, just so that the brain can sort information out, so the brain can sort information that's coming in from the visual world and so forth. All of these things have to happen in a form. The sorting has to occur in an appropriate form, and of course, it does not commonly in the autistic child. And the result is impairments in the social cognition domain, and ultimately in domains of social control. So things have to go right in this period. They don't always go right in this period. This is a period of riotous change in which all of this initial sorting has to occur. And if it occurs in the appropriate way, the brain is set up to make use of this information in its more complex operations. Now, moving forward from this period, of course, I'd say across this first year of life, that brain plasticity has been all about creating the person. And by the person, I mean the operator in the world. It has to advance in all of the ways that make it successful in operating in the world. It's also creating the person, the self. And again, this is occurring through massive self-reference, billions of times in the life of the brain, in the life of the child. The brain is associating the feelings, the actions, the thoughts with the source of those feelings, actions, and thoughts. And of course, that's to the brain itself, you could say to the person itself. And by its nature, commonly, the brain is going to associate the operations of that person with things that are close to it, and things that are close to it under conditions of positive reinforcement. So it develops an attachment, of course, to those things. They actually grow into the person that is the developing child. That you could say the mother is part of the essence of the person that is the child, just like the baby is the essence of the mother, the person that is the mother. We're built for attachment. We're built to be attached creatures. We're social creatures. And basically, this is a critical thing that has to happen across life, that is important to happen across life. It's a critical thing that many children in the autistic spectrum struggle with, we know. At an older age, plasticity is regulated. So the brain initially does not regulate plasticity, you could say very, very directly. But later it learns, it matures the machinery that's going to control when change is permitted as a function of outcomes, and it's a function of the context in which the brain is in operation. And by this regulation, it only permits change in a sense when it matters to the brain. And change is controlled by outcome. That is to say, whether the outcome is judged by the brain to be good for it. Now, a very simple example of how the brain changes or specializes can be understood by understanding how it might specialize to acquire a complex skill or ability. For example, the skill of reading. If we look into the brain of a child that's in the act of reading, we see that there is a reading brain in place. There's actually machinery in the brain that's engaged that, of course, is not engaged in the same way in the non-reader. You could say the brain has specialized in its operations in all kinds of ways, and we can record innumerable ways in which it's operating in a different way. As this complex skill in which the animals are relating the sound parts of words that bear meaning in the words to these little scribbles, the orthographic or visual representations of letters of words, in these long strings that provide us a basis with fast, ongoing translation. And this is an incredibly complicated skill that most children master. Of course, many autistic children struggle with it or struggle to express that they're capable of it. Many more have the ability to do it than they're able to tell us about it. But we know that a child that advances in the skill actually develops a reading brain that's different, that accounts for the skill. So whenever you acquire a skill or ability, you're actually remodeling your brain, and that remodeling accounts for the ability, of course. The brain is continually changing itself in thousands of ways. It's revised itself. And the sum of those revisions is that evolving person that's evolving in ability as you advance in life. And of course, one of the things we want to see in every child is a wide and broad and deep improvement or mastery of abilities that empowers us in life to become an effective older child and adult with a powerful operating brain. Now, of course, many children struggle in different ways from genetic and from environmental reasons. And at an older age, we can find that they might have the brain of a non-reader. And that's really what's expressed in these images on the right. We distinguish readers and non-readers. These are actually images from 20 children that are normal readers on the top, on the right, and 20 children that are non-readers on the bottom. These are all children that we identify as being dyslexic on the right. And what's indicated by the green circles are areas that are engaged, that are specific to reading, that this more posterior area, which I'm going to indicate by an arrow, we know is a zone in which the translation of word sounds to letters is being achieved. And this zone more anteriorly actually relates to the identification of the meaning of words in reading from the written script again. And this all relates to the rapid interpretation of this sound to orthographic or visual representation of sound. And we see that there's a complete lack of activity in these zones, or very disorganized activity in this non-reading population. We could look elsewhere in the brain and we could see other differences, of course, that relate to other dimensions or aspects of the reading art or reading ability. Now, it turns out it's quite easy in the great majority of children that are non-readers to train their brains, even though they might have inherited weaknesses, even though they might have lousy childhoods, to recover reading ability, and as you recover reading ability, to reorganize activities in brain so that these children again have a reading brain. And we see that the images from 20 children shown on the left that have gone through such a training program or strategy. This is a computer-delivered strategy called Fast Forward. And it turns out that 17 of 20 children that were trained with this on these tasks over a period of an average period of about 23 or 24 hours, reacquired the ability to read. The ability to read. Non-readers now can read. It turns out that 3 were resistant to training for perhaps their genetic faults or whatever, for whatever reason. Basically, they could not be driven plastically to correction. Now, you might be interested to know that this training did not involve reading. Actually, what it involved was the training the child to more accurately sort word sounds. You could say it's like if we're going back in a baby again, and helping the brain more normally, or you could say more accurately, sort those phonemic inputs, those sounds that are the basis of phonics reading. And now the brain is doing that more accurately. And now to this child with this neurological correction, reading makes sense to the brain again. And reading can be accomplished or achieved in these older age children. So these are children between, I think, about 11 and 13 years of age. So the brain can change. There are strategies by which you can change it. The point I want to make is that the change was not driven, it was driven by making a deeper consideration of the underlying distortions of the neurology. And so this applies for any neurological correction. When I look at what a child can do or can't do, I have to consider how all of the assets that must be brought to bear to account for that behavior must be addressed by correction, if correction is required. And this is an example, I hope, that makes that point. So I want to go from this point, understanding that our brains are continuously changed and those changes account for our evolution of our abilities, to talk about a key aspect of adult cortical plasticity that's illustrated by the example I've just provided, and that's the indication that these underlying brain processes are always advanceable, and in fact, whenever we see regression or whenever we see a brain that's limited in its performance, the processes can be shown to be reversible. Now, to illustrate this point, I want to talk about an experiment that was led by a wonderful young French-Canadian colleague who's now at the Montreal Neurological Institute. His name is Etienne de Villers-Sedani. And the experiment involved looking at the changes or the differences in the brain between the rat in the prime of life, and we see an animal in the prime of life in the upper left here, versus an animal near the end of life. And we can show that he's near the end of life, for one thing, because he has white whiskers and he has white paws. This is an animal that's expected, you could say, to pass from this mortal coil within a month or 2 or 3. And what we're going to do is look in the brain broadly. We see that this animal is now sluggish, and we can demonstrate that he's cognitively limited or she's cognitively limited in all kinds of other ways. We've also done equivalent experiments in most of the things I'm going to talk about in young versus older humans. Now, what we see when we look in the brain, first of all, broadly, is just about everything we look at neurologically is different in an animal near the end of life versus an animal in the prime of life. And you can say, well, how many of those things that are different really advantage the older brain? No, the simple answer is none of them do. So every way you look at the brain of the older animal is deteriorated. It's demyelinating. It's losing its insulation on its wiring. It's synaptically simplifying. Its connectional sophistication is declining. It's shrinking. There are chemical changes in the brain. The brain is operating much more slowly. It's operating less precisely. In almost everything you look at, there are very clear large-scale differences between the old versus the young brain. And now we ask the second more important question. Which of these differences can be reversed, partially or completely reversed, by specific forms of training? So what we've done is we sent old rats and many, many old humans, hundreds of thousands of them, you could say, to the rehab clinic. And what we're asking in these studies are, how many of these things that are different in old versus young can we drive correctively? How many of them are reversible? And the simple answer is all of them. Everything can be driven in a corrective direction. So here are a few things that we've looked at. We looked at myelination, or the insulation on brain wiring. We see that locally and in brain systems, myelination is substantially recovered, and a relatively limited course of training over a period of about a month. In the systems that we engage, most of our experiments were done in the listening brain. We see the listening brain is substantially remyelinated. We see that local brain cell teamwork or response coordination is restored. And this is a critical aspect of brain function that's expressing how well the brain is sorting information. And we see that this sorting is more powerful. We see that the brain is operating in a more powerful, locally coordinated way. It's picking off information in a way that would express greater functional reliability. It's also increased the coordination across systems and between systems. That's recovered. Now, very importantly, from the point of view of reversibility or the ability to change processes in autistics, we see a recovery of inhibitory power. So we see very gross degradation of inhibitory power, and it's differentially affected in aging, just like it's differentially impacted in autism. We see that it's actually substantially recovered in old trained versus old untrained animals. The neuron numbers are back. The numbers of neurons are labeled for inhibitory cell markers. The cells are big again. The synapses are big again. Everything about the vigor of the responses is recovered. The responses are fast again. The brain is also recovering the receptors, these chemical receptors that are accounting for the characteristics of operations on the excitatory and inhibitory processes in the brain. Those change in a negative way in aging. It's recovering its speed and its temporal representational fidelity. So it's operating at speed again, and it's operating so that it's representing the details of what's going by in fast time with high accuracy. This is critical for things like the detailed language operations of the brain. It's recovering a whole series of other critical things that relate to its ability to control itself and its operations that are further down this list. But what's important is that everything we measure is corrected. Now, this is not an accident. You know, how many of these things would we have had to look at before we can come to the conclusion that the brain is constructed to be reversible? It's fundamentally reversible. So what is that all about? What does that mean? How could such a complex set of regulatory processes and this complex interaction or play between genes and the physical and functional and chemical brain, what could it be all about? Now, another simple question we would ask in this class of experiments is, well, if it's reversible, then that means we ought to be able to take a brain in the prime of life and turn it into a brain that looks like an old one. It turns out that's very easy to do. All you have to do is to add noise or chatter into the processes of the brain. And if you do that on a significant scale, within a matter of weeks, the animal looks like it has the brain of a very old animal. Now, when we look at the brain of a very old animal, a brain of a young animal that's now being advanced to look like the brain of an old animal very rapidly, we begin to realize, because these processes are reversible, realized several years ago that we were also looking at the characteristics of the brain that had had almost no experience. Very old in a brain is an awful lot like being very young, because the processes in place are fundamentally reversible. You can think of life as like a kind of roller coaster ride where you begin with a very high noise, very disorganized sort of condition, and in the normal case of development, you roll into more and more powerful local organization. Now the brain is sorting beautifully, and it's using this information and powerfully manipulating and burning in all of these abilities through experience-related plasticity. And then noise creeps into the machine. The machine begins noisy and begins to chatter a lot. And as that occurs, basically the brain goes right back in the direction that it came from in these reversible processes. And ultimately you have a brain that's more and more like the brain you started with when you came onto the planet. Now, why does the brain do this? That's a really interesting point. Now, let me just talk a little bit about brain plasticity across the lifetime, and I'll try to explain that to you in simple terms from the point of view of, I'm talking about our perspective. So first of all, understand every brain is fundamentally plastic. They're constructed to change. They have to change. They have to evolve. And every brain, including every brain of every child with autism or any other impairing or disabling or condition or adult, has a brain that is on some level plastic because they've evolved in their ability, they've changed in their ability, and plasticity accounts for that, underlies it. We all have this resource. That's not to say that it's not compromised in some of us, but we all have it in there to help us in ways that can enable us conceivably to be better, stronger, more capable tomorrow as opposed to today. Of course, every brain also has a different plasticity experience. It has a different history. We're all massively, individually, idiosyncratically specialized. I once had a conversation with the Dalai Lama, and I said to him something like, we now understand the basis upon which we create the specific abilities that we define it. And he said, well, maybe so, but you can never understand or interpret the differences that account for each individual self or person. And he's right, of course, because they're based upon billions of events of change occurring in your interaction with the world that only you pass across in the same way, no two people alike, combined with billions of events that occur internally within your skull that are unmatched by any other, can never be identical with any other. You're a unique person because of this plasticity. Every one of us has a different plasticity history. And in a sense, a child that struggles, an individual that struggles with autism is another individual that has a very different plasticity history. We can think of them as different, not as worse, not as better, not as any such a thing. They're different. It's different, just like we're different from every other person. Plasticity, of course, is good for life. You can't kill it. It's good for life. Well, maybe in the very end of life, it can fade and become almost useless. But up to that point, it's in there because we know at any point in life we can improve our ability, we can change things. Brain plasticity is by its fundamental nature reversible. And the reversibility is all about the brain self-regulating its operational machinery. What the brain is up to is it's advancing when it can. It's going faster. It's making more complex judgments. It's improving its operations when it can. And what determines when it can? It has machinery in it that's controlling plasticity that's all based upon the brain, in a sense, getting the answer right. So as you improve, the brain knows. It's evaluating that, and it's determining when you're improving. And when you're improving, and as you're improving, the brain supports that improvement. But when you don't get the answer right consistently, the brain has to regress. And the reason it has to regress is because now the brain is not rewarding itself for not getting the answer right. Basically, it has to readjust its machinery so that it does get the answer right. So the brain basically has a wonderful set of processes in it that assure that you always are connected in a way that can support your operations and, in a sense, help ensure your survival. And it's actually constructed on this basis, constructed with inherently reversible processes. Now, what we're going to talk about in a minute is using that reversibility to drive changes in a brain that's struggling. And that's what we're trying to do in many individual populations, including the populations of kids with bad childhoods, from a brain bad child or difficult childhoods. The brain is establishing order. It's generating correlated activities in incredibly complexity that establish the cortical network processing by its little functional units. We call them mini columns. It's organizing all of this activity from noisy chaos. And this is an incredibly complicated and serious challenge for many young brains. We know this. So it has to do all of this sorting. It has to do all of this organizing in an effective way to set up and empower its operations in later life. Then finally, I've just described how process noise can either not be cleared up, you could say, because of the way the brain is organizing itself efficiently, but it can also grow, commonly does grow in the brain of the average older individual, and in many other clinical indications that we acquire. So whenever the brain struggles, it changes its operational characteristics. It goes downhill, you could say, to some extent. And that growing chatter or internal noise is substantially underlying age and condition-related decline. I might just say one other thing about that is that scientists have looked at gene expression, that is to say how the genes are changing the chemistry of the brain as we look at how the brain changes in psychotic illness or in a variety of neurological conditions. And what one sees is an incredibly complicated set of genes that are upregulated. That is to say, it's producing more of this chemical and less of that, and different constituencies are changing and so forth. And this was thought for a long time to be providing a great source of insight into the specifics of, let's say, schizophrenia or autism or whatever. And we now know, in fact, that to a large extent these genes are actually simply expressing this adjustable and reversible plasticity, because incredible level of gene regulation has to occur to drive plasticity in a forward or negative direction. So from this perspective, what goes wrong in autism? Well, first of all, inherited weaknesses, we know, are degrading the processes that control plasticity and development. So this is a common thread that we see indicated by the beautiful recent studies that relate to both the genetics of autism, and these studies have been very, very crucial in our understanding, and also to our seeing the expression of differences in the autistic brain as opposed to children or adults who are developed quote normally, end quote. And we also know that there are processes that impair cellular and network the machinery that's controlling chatter in the brain. I can look at a whole series of processes I know that are contributing to the brain sorting information reliably and operating under control reliably in this sense. And a whole set of processes are going to contribute to increased noise in the machine. And in other ways, there are many other things that impact plasticity in ways that can degrade or slow early plasticity-based remodeling. That is to say, can frustrate the brain from getting the answer right in the domains in which the brain is trying to move or advance. And many things can frustrate those progressions. So there's a combined set of factors that relate to these, that come substantially or relate to these inherited weaknesses that are contributing to problems. And this is one of the reasons I think that the genetic picture is so complicated. Lots of factors can contribute to making the brain noisier and to contribute to its struggles in organizing itself in this early life period. Secondly, we know that there are environmental factors that can be expected to add to the chatter and to further degrade or slow plasticity remodeling. I'm going to talk about some of those in a minute. And we know that if we add that burden, just add that burden to the genetic burden, we know we have bigger problems for the developing brain. The brain's struggling anyway. And let's just pile on. Let's just make things more difficult for it by adding to other factors that are going to add to the intrinsic noise in the process of the brain. Now, the brain is going to it's going to do the best it can to get the answer right. And it's going to advance to the point where it's now controlling its plasticity and change, even though it's sorted information perhaps in a very different way in autism. And the brain's going to adopt degraded operational characteristics because that's the best way it can operate to get the answer right. Now, one of the problems with that it has is that because it's so noisy, because there's so much chatter in the machine, it's subject to seizures, and it's subject to sharp stack activities, and it has other problems that are going to help sustain that high noise condition. Now, we can do things that can offset or overcome that can contribute to clarifying things again in it. But I'm going to say that these things are going to be contributing to the problems of reversing the processes that can drive it into the clear sky again. So this is what I think from a brain plasticity perspective is in play in the genesis of autism. Remember, this is a scientist's perspective, and I could talk about this in a lot more detail. And remember that it's a sort of extended or exaggerated hypothesis, and a lot of things about these processes, of course, are unknown. Now, the result of all of these things, and many other contributors could be in play, are persistently noisy cortical processes and degraded cooperative processing in the brain. Brain is not operating with the normal level of coherent operations, and because it's not, there are relatively poor mini-column formation and relatively poor limited response coordination. There's elaborated non-selective cortical network wiring. We know this. We see this in these brains. We see it recorded in autopsy. We can record and see it manifested in various ways in the brains of autistic children and adults. We know that the autistic children, as a consequence of this, have relatively non-selective attention, prediction, and memory association processes. Now, they can have incredibly powerful memory processes. But basically, the problem is that when information is not sorted, sorting is critical for controlling sequencing and prediction and memory association. And we know that commonly this represents an inherited weakness of a brain that has struggled to organize itself down this path. We know that the brain is inherently unstable, and we know that it's susceptible, and in a large proportion of these individuals will see evidence of that susceptibility to seizures and to sharp spike activity. And seizures and sharp spike activity are a source of continuous noise. They're basically frustrating the advance of the brain in sorting information and renormalizing itself. We see degraded and stable regulation of the neurotransmitters that are actually controlling aspects of learning and change in the brain. Serotonin, noradrenaline, acetylcholine, dopamine, their modulation, their regulation are all affected. And the reason they're affected in part is because their control of the release of these neurotransmitters are determined by outcomes, but they're also determined by and controlled by feedback that's coming from the brain. And because the brain is relatively disorganized in its activity or organized in a very different way in its activity, that regulation is not normal. So we have the child that struggles to control the levels of attention. They're either incredibly powerfully engaged in the expression of noradrenaline, which is controlling their alertness or brightness, zeroed in on something with incredibly overpowering forces, or the machinery is off, almost as if they're in a state of complete inattention and disengagement. So this is a problem of regulation of these processes because of this abnormal progression in the brain development and control. And of course, we have an inherently unstable brain. An inherently stable brain can struggle to categorize, associate, predict, and attend. And we need to do those things, of course, to relate vision and emotion and mood. That is to develop social cognition and control, and also to develop things like phonemic reception to meaning of physical action. That's oral language, that's talking. Many other things like this require these associations, require this categorization, require this prediction and predicting and intending power. So this is the consequence, I think, of this, the predicted consequence of this developmental progression or history. Now, we've also looked at environmental factors, you could say, that can amplify the brain struggles in development. I'm taking a little longer to talk about these things than I wanted to, so I'm going to talk about this very briefly. I became very interested maybe 12 or 13 years ago about the origins of the apparent increase of the incidence of autism. And I thought somebody should begin to go down the sort of obvious list of factors that we know are changing in incidence in modern environments that could contribute. So I've looked at 6 of them. That is to say, scientists that have worked with me have looked at 6 of them. And what we've discovered is that on all 6 things we looked at, the environmental factors in play would almost certainly, almost without a question, make things worse in a brain that is inherently noisy that's struggling to organize itself. So among those things, we looked at the consequences, all of these in levels of exposure that we know occur in natural environments. None of this is extreme. All of these are animal models, all of these things done in rats. So the rat is administered PCBs or PBDEs. We either give it to the mother or we give it to the baby rat. And later we look at the consequences. And the consequences are a grossly degraded processing of information in the brain. We can look at it at the way it adds to a second burden, so we can raise the animal in another way that gives them a difficult childhood, and then we can add the PCB, and it makes it all the worse. We've looked at heavy metal poisoning, same thing, grossly degrades the way the brain processes information. We looked at noise exposure in infant environments, same thing. Raise a kid and lock in the den, and it's the brain, of course, you're adding the noise now extrinsically. You're not directly in the machinery of the brain, but it makes the brain noisier, full of chatter, and it adds to the den, adds to the problem. We've looked at the consequences of early cord clamping because we know that that's something that evolved relatively recently. People are being more sensible about this in the last 3 or 4 years, but we know that deprives the brain, the blood of iron. We know that has consequences in oxygen supplies in a compromised brain. Not good. We see it having consequences. We looked at perinatal asphyxia. We know that many more children are born early and then variously struggle in oxygenation early in life. Has big consequences. We've looked at trimester ultrasound in high-res modes. Repeated very clearly distinguishes those brains from brains of children that haven't gone through that procedure. It's not good. We looked at SSRI medication, known to increase the incidence of autism. Well, it also degrades the operations in the brain and so forth. All of these things can be expected to add to a young brain's inherited burdens. And, of course, many other environmental factors are on this list. So when people ask, well, do we know what accounts for this increased incidence? Yes, we do know. How many of these things do we have to look at that we know are changing stronger now in contemporary environments than earlier, 30, 40, 50 years ago, before we're pretty sure that the cause of this, like global warming, is we humans. We're doing things, in some instances, recklessly, in other instances, not thoughtlessly, that are going to contribute to this burden. And we can expect these things to grow as problems in human populations into the future unless we do something about it, face up to this. So in these class of problems correctable via brain plasticity-based therapeutics? Could we actually apply brain plasticity to drive a correction? Well, we know that there are children that have remarkable recoveries from autism. We've seen this in our own populations. So I might say using the training programs, the language training programs that we've developed called Fast Forward, we've actually trained tens of thousands of children in the autistic spectrum. And among those, we've seen children that have almost miraculous recoveries or improvements. Now, I don't want to make too much of that because the great majority do not have changes on those magnitudes. We see substantial improvements on the average, but we also see if we train 100 children, most children get better, some children get remarkably better, some children show almost no positive change at all. That's really interesting, but that's what we see. We know that there are miracle children, though, you could say, and these have been described in different forms. So we've seen children, well documented in our own trials, that have moved on standardized testing 5 or 6 standard deviations across the distribution, across a relatively short period of time. That is to say, seem very autistic, and then 3 or 4 or 5 or 6 months later, seem very normal, even exceptional. Now, this is a very unusual circumstance, of course, and nobody can imagine that this could happen in their child, but we know it can happen. Now, those children moving to that extent across the distribution over that critical period of time, we're not changing the brain in all of the ways that empower them to see the miracle. You have to say they had tremendous potential in there, and there were sort of linchpin things that must have been holding them back. But we know that the brain is plastic, and we know through lots of good brain work, we ought to be able to drive improvements in almost any ability that's impacting or limiting the child. Now, we also know that from animal studies, because we've conducted many studies in animals where we've given animals a very bad childhood, you could say, in all kinds of ways, and then we've trained the animal. And I might say that in every one of those studies, so for example, we've given animals the kind of burdens I talked about just before, and in every one of those studies in which we've applied training in the model, we've driven the brain correctively, so the brain looks like a normal brain. Now, I want to describe a little bit of one of those studies, and this is a study conducted in collaboration with Xiaoming Zhao, who's at the East China Normal University in Shanghai, and with Rick Lin, who's a professor of anatomy at the University of Mississippi Medical School. And in this very complicated next slide, I want to talk about broad-scale recovery we see in animals that have been, in a sense, poisoned by giving them a dose of an SSRI. So this is a standard medication given might be given to a pregnant woman, to prevent depression or overcome depression. Very important to treat depression in a mother, pregnant woman. Absolutely critical. But it turns out SSRI has consequences, neurological consequences, because it affects serotonin regulation dramatically. That's what it targets. And when we look at it in the behavior of an animal that's born of a mother that has had such treatment, we see consequences. We've documented those consequences behaviorally. We see the animal is struggling, the brain is struggling in different ways behaviorally. And then we train the animal in a very simple way. And again, the animal is working for about an hour a day, in this case for about 40, 45, 50 days. And what we see is the animal's brain is almost completely recovered in a functional way, looks normal behaviorally. And in a physical way, we see very substantial positive correction. Again, all of these processes are, by their nature, reversible. So now we've given a rat, in a sense, a very big burden in its very early childhood by basically poisoning it through this in unusual way. And now we're looking at the consequences of training it, and we find we can substantially train the brain out of it. And that's what all of these complicated slides show, and I'm going to skip them because I'm running low on time. And I'm just going to show a couple physical changes. Here we're looking down into a nucleus in the brain called the dorsal raphe, and this is the forebrain's main source of serotonin. And you can see that, perhaps it's a little bit difficult to see, but you can see that by looking at the top panels, there's a significant recovery of the expression of serotonin in this brain of the trained animals, not all the way back. This is the control of the normal. And this is an animal that has received the poisoning of the SSRI, but basically are untrained. And these are significantly different. And you can see these differences expressed in the bottom. These are all of the different divisions of the source of serotonin in the brain. And the green bars are the trained animal bars. And you can see that in general, they're all higher than the red bars, which is the untrained animals. So basically what you're seeing, I'm sorry, it's the other way. And low is good here. Sorry. You can see that training always advantages the brain. So here's the second example. I might say that we also have seen this in other physical measures of response in the transporters. And these effects are even stronger in the expression of dopamine, another key neurotransmitter, and the expression of noradrenaline. In inhibitory neurons, I'm going to show you an example of that in a minute, and in excitatory inhibitory receptor subunit ratios. And we know that an imbalance of excitation and inhibition, we think, is elemental aspect of the distortion that occurs in autism. So here's the second example in which we're just looking at inhibitory neurons. Here we're looking at a trained animal brain, a section of trained animal brain. This is a typical untrained animal. You see the label cells are sparser, very significantly sparser. And here's a naive animal, that is to say, a normal animal that's never gone through the poisoning. So this is more like this, and we can see significant consequences that have been recovered from by consequence of the simple form of training. Now, how do we approach remediation in children and adults with autism? Well, first, what we've been trying to do with the support of the Simons Foundation is studies in which we're assessing the deficits associated with the specific form of autism that comes from a gene deletion, the so-called 16p11.2 deletion syndrome. And our strategy has been to try to, first of all, determine what's wrong in the child neurologically. And we do that by applying a complex assessment battery. And we look at a whole series of affected domains which give us an insight into the affected brain systems. Basically, what we're trying to establish in this child population is what's wrong, you could say, and how definitively can we define how we might approach repairing it by intensive training that's neuroscientifically based on the basis of brain plasticity. So what we're assessing are the operations of the brain in attention, in memory abilities, in executive function, or the ability to manipulate information in complex ways, in processing speed, in social cognition, in auditory perception and language abilities, and visual perception and complex visual abilities. So we're basically looking broadly at the brain's operation in all these domains. We actually have a 38-point assessment strategy. All of the assessments are delivered in a game-like form on the computer. It takes about 70 minutes overall for a child to get through these assessments. The great majority of children in preliminary studies appear to be able to complete this successfully, including children that are substantially impaired in the autistic spectrum. And these are just an example of what these game-like assessments would look like from the point of view of their gameplay ability. They're actually designed, you see some writing on these, but they're actually designed to be played and to be initiated in the child that cannot read and that does not read. So it doesn't really require the child be able to read in order for the child to operate or complete these games. Now, when we look preliminarily at these kind of exercises, we find that through an assessment that's applied just over a period of 2 or 3 or 4 minutes, we can actually pretty nicely gauge the abilities that a child has in particular assessment components. So here's an example of an assessment in which we're looking at the child's ability to match faces. Or here's the ability of the child where we look at the ability of the child to look at the position of the eyes. And basically in these simple assessment tasks, we see that distinguish between children that are normal, low is good here, versus children that have ADHD, less impaired than children that have autism. And we can see these distinctions. This is obviously a neurological target for correction, as is this for gaze matching. In the right, up is good. That is to say, these bars for the normal children being higher are good. And you can see, again, they distinguish these 3 populations. You can see this is a measure of your ability to, this is control baseline levels of arousal. And this, again, is a face matching exercise. Now, we know that behind every one of these 38 exercises is a strategy for correction. And basically, we can correct these abilities if we looked at them individually. And here's some simple example where the child is exercised to identify emotions in an emotion naming task. And now we train the child in a relatively limited way, in ways that we know will drive improvements. Emotion naming, we see a recovery. Face perception, we see a recovery. Eyewear, and again, this is indexing the recovery. So our whole idea in working with the Simons Foundation is to devise a strategy, to develop a strategy by which we can more completely understand the specific deficits in an individual autistic child or adult in front of us, and then on that basis, basically marshal our brain training resources to drive the brain correctively as completely and as rapidly as possible. So to sum up a few implications. Now, the first thing I want to say is that I think that autism is an expected final common pathway of aberrant development. There's nothing terribly mysterious about it. That has a myriad of contributing genetic and environmental causes. There's mystery there because we don't understand all of these influences and factors that are in play, but we know a lot of them. And we know that ultimately they carry the brain down this alternate path of development, and they create an individual that has these substantial differences. Not worse, not better, different. We know that autistic brains are plastic. We know that sometimes that plasticity is compromised by the genetic faults. That is to say, they're going to struggle a little more to learn or improve. We know in other instances they don't seem to be. It doesn't seem to be that impeded. But they can all of them can at least usually be driven in corrective directions by appropriate training. Now, all is probably too strong a word. It is too strong a word. Most can be driven, we think, in strong corrective directions by appropriate training. We have a growing understanding of what to correct, and we're trying to become increasingly more sophisticated about that in ways that could allow us to correct a specific child's neurology with a consideration of their faults insofar as that is possible in any given individual. Training should target neurological renormalization. That's what we're trying to do. We're not trying to recover the behavior of the child. We know if we recover the brain neurologically, the behavior will be recovered. So it's beyond addressing deficits in expressive behavior. We're not trying to socialize a child by talking to them about what social behavior is about, or train a child to compensate. It's not about that. We're trying to correct the neurology so that that socialization and that compensation or that recovery occurs. We're trying to recover the brain and not compensate for it. For some forms of autism, training is facilitated by drug-achieved recovery of plasticity. It may be a crucial aspect of effective treatments. I might say that Simons Foundation has supported several wonderful scientists who have identified potentially powerful and useful drugs. And they address issues that relate to plasticity itself. And you certainly want to fix that if the drug can compensate for that or correct that. And I think that in combination with the use of those drugs, training can be a key. Ultimately, the brain has to go through a training progression if we're going to see a relatively rapid and relatively complete renormalization. And finally, I think we should all be sensitive to the fact that some forms of autism may resist large-scale recovery because there are factors which are not addressable by training or drugs. So there can be physical anomalies. There can be differences of the brain. The blood-brain barrier can be compromised in ways that are now currently irreparable. There can be other things that will frustrate complete recovery. That's not to say that we can't always go some distance in a corrective or improving direction by training, and maybe by training applied with drugs. And so I think that we have all increasingly stronger prospects for doing more to help these children and to help their families. And it's one of the great missions that I have in life. I hope before I'm done, I can make a more direct and complete contribution to this. It's something that I know is incredibly important to do for our societies. Thank you very much. I want to say one last thing, and that is I've written a book that with supporting scientific documentation at a website. And if anybody wants more information about the substance of this talk or wants to go to find the scientific references that support what I've said, it's not specifically about autism. It's about the plastic brain in childhood and adulthood. But you can find a lot of information that supports these things there. So I'd strongly recommend you go. The book is called Softwared, and you can easily get it on Amazon. Thank you very much. Nice to talk to you.
Okay, thank you very much, Dr. Merzenick, for sharing a very intriguing perspective with us today. Although we're running a little bit over here, I want to try to squeeze in at least one or two questions, and then we'll have to move the rest of the questions online when I post this replay. Pascal Wallish from David, a postdoc at David Heger's lab at NYU, he wrote in, and he wants to know, I think, based on your description of critical periods and self-genesis at the beginning of your talk, right. I guess at a conceptual level, would you describe autism as essentially a failure to generate the self?
Well, I think it's weak. It's altered in that sense because it's weakened in its associative powers in a kind of organized way. It's not that it hasn't developed, not that it has no capacities, but the indication is that it's not going to be so powerful or organized or coherent, and I think it's got to be in play. I mean, we know that the associative abilities are going to be weakened in a sense because the brain is creating a construct of the world in which it hasn't categorized things in the normal, reliable way, or with normal reliability. I mean, there's power in that. Power is that it's not in a rut like the rest of us, always leading ourselves in prediction to the next highly expected thing. That's the way we are, that non-autistic is. Autistic, in a sense, has the freedom to move their thought anywhere. But also there's the burden. And one of the things that means is that they're not so strongly and reliably associating those things that are happening in their brain, you could say, to themselves as the source. That's my belief. Okay.
Another question that came up a couple times throughout the talk, I'll try to summarize them or make a consensus question out of it. In relationship to noise, when you use these terms noise and chatter, in particular when you talk about turning a young brain into an old one, just add noise. Now, when you say noise, can you elaborate on that? Do you mean physical auditory noise, or is it more cortical noise, systems level noise, or both?
I mean cortical noise, or you could say systems level noise. You could either sense because it'll get to the cortex. We've actually accomplished it in two ways. We've actually applied acoustic noise because we know it makes the listening machinery of the brain inherently noisier. So if all sound comes in against a background of noise, you could say then the machinery is always continuously noisy, and that results in a rapid maturation because of the noisiness of the machine, you could say. But also we've increased noise by manipulating inhibition in the brain. And when you weaken inhibition in the brain, the processes of noise are just inherently noisier. There's more activity that's disorganized and non-contributing. And this also will result in the same set of adjustments. So I really mean noisiness in the machinery of the brain. There's just lots of activity that's occurring that is meaningless to the brain. It's just chatter. It's not part of the useful organized activities of the brain. That's the problem. And if you're getting rid of that when you are engaging the brain in ways that keep it basically sorting and keep it in its highly coordinated normal operations. And to the extent to which you're not doing that, it's increasing this internal noisiness.
Okay, I see. When you were referring to some of those studies with the SSRIs,
right.
and you showed the rescue for
right.
animal through the training with the rodents and the animal training, could you describe just briefly what exactly the animal training was in some of these cases?
Well, the animal is making distinctions about the time modulation of sounds or about the frequencies of sounds. We've trained animals in several ways and driven these positive changes in which the animal is basically progressively initially making relatively crude distinctions and ultimately making those distinctions in a more and more refined way. So commonly the animal is trained to distinguish sounds that are modulated at very different rates and ultimately in a refined rate, or distinguishing differences in the pitch or tones of sounds that are initially far separated and ultimately very refined. It's doing this always against the background in which it's challenged to pick out sounds against the background of many other sounds. So, and these forms of training have turned out to be really important from the point of view of driving these corrections, and they're implemented in the strategies that we apply and shall apply in autism and in other challenged brain conditions. They're relatively simple, surprisingly simple in form, and yet they have broad-scale, relatively profound effects. I might say also, we don't just see the changes in the lower levels of operation of the brain where you think might be impacted. We also see recovery in the frontal cortex. We see changes in the hippocampus. We see changes broadly across the brain.
Okay, we'll try to get to one last question here. This is from a viewer today who wants to know, could you summarize again in the preliminary studies you've done in populations of people with autism with these brain training platforms, how many hours, generally speaking, how many hours of training and over how many days before you see progress in these
right.
studies you've done?
Well, if you train a child we've published studies and that have been applied, for example, and other people have looked in studies in which they've applied training like the training in Fast Forward, which is a language listening training program. We've conducted similar studies in which we're trying to improve, for example, social cognitive abilities in children. We see significant effects within, 5 or 6 or 8 or 10 hours of training, but of course those impacts are incomplete. If I'm trying to reconstruct, you could say drive the social cognition training abilities of a child as far as I can towards normalcy, it might be appropriate in the brain training side to apply training that might be extended over many tens of hours. So, in other words, how much you train is a function of how completely the consequences and how extended the consequences. In social cognition, we've actually created 24 different training modules that relate to different aspects of social cognition because it's complicated. Similarly in language. So of course you can see some benefits relatively soon, but the brain has to go through a lot of change in order to really recover their language. It's to recover its language abilities, or its ability to talk fluently and with high fluency, or its ability to operate with high facility in social cognition. And also these operations, of course, Greg, have to be extended to real life. You have to also operate in the world as a social being and and in your language operations.
Well, that segues well into kind of my final question is how the big question in a lot of these kind of task-specific training trials is how generalizable do you expect these improvements can be? So, for instance, are you just training individuals to perform better at these tests, or how scalable do you foresee these improvements might be? And maybe we'll end on that note.
Yeah, that's a wonderful question, Greg. And the essence of the answer is, if I train an explicit ability, so I have some, you could say, higher level ability that I know is impaired in a child, I can have the child practice that, and I would almost always see improvements. But I haven't really changed the system that accounts for operations in that domain. And unless I change the system, I'm not going to have a generalized impact. So actually, we are always trying to change the system. We're trying to change the whole machine, not just some ability that springs from the machine. You have to change the operational characteristics of the whole machine. And that's our approach. That's a neurological approach as opposed to a strictly behavioral approach. You have to think about how you can correct the brain, not just some list of things that you relate to the behavior. So I think the more we understand how to change the brain, and then what we've done in every trial that we've conducted or supported is we looked at the extent to which it generalizes. We're a failure if we don't see generalization. And what we see over and over and over again is that it does generalize. Things that aren't trained in the domains that aren't trained, including things that are far removed in so-called far transfer effects, we see improve. And that you can expect if you've actually corrected the neurology of the brain in this more complete and fundamental sense. So it is possible to drive these changes in ways that generalize in a powerful way into the daily life of the child or the adult.
Okay, well, it looks like we'll probably have to end things there. Dr. Merzenick, thanks again for a really thought-provoking presentation today. For those of you tuning in, a complete replay of today's session will be available on our site at safari.org. I'll try to get that up later today or tomorrow at the latest, and you'll all get alerts in your inboxes when that posts. Safari webinars are a monthly series, so check the site for upcoming sessions, as well as if you want to watch an archive of previous talks. A good way to keep up on that is to join our newsletter if you haven't already. And once again, I'd really like to thank Dr. Merzenick for sharing his remarkable work with us, and I'd like to thank you all for tuning in.
It's been fun to be with you.